We love our museums here on Tested, and one of the most important and august museums in the country is right here in San Francisco, our own California Academy of Sciences. It was founded over 150 years ago and is not only a world-class museum, it is still an active and important research facility. I recently visited their Institute for Biodiversity, Science, and Sustainability, the Academy's research arm that is home to over 46 million specimens from all over the globe. I visited the backstacks of the Academy to learn the history and the care of these amazing collections and how researchers are scanning the collection digitally with modern scanning technologies, all to help the public better understand evolution and the interconnectedness of organisms and ecosystems.
Kate, this is your scanning setup, is that correct?
Yeah, one of the scanning setups that we have.
This is a Galapagos tortoise.
Yes.
And you are about to show us how the scanning works. Now, this is how you scan large specimens like this.
Yeah, we actually went through a couple different setup trial and errors, and we came up with an inner tube as a buffer system or a bumper system because in order to get the underside of the tortoises, we needed to have enough padding.
Right.
And this allows us to kind of control how much air goes in. So the different tortoise shell shapes, the different sizes. Yeah, exactly. So that's kind of how we ended up on the inner tube.
And this works both right side up and upside down?
Yeah, we actually don't need the inner tube when we're doing the right side up. It can just be on a table or a flat platform, and we can get the majority of it. So we only really do the upper side and then flip it over and do the underside for the tortoises.
Now, is this a completely preserved specimen, or is it like taxidermy or some mix of the two?
Yeah, some of them are completely taxidermy, some of them are plastered, and then some are mixed. As you can see, like some have metal rods in there.
I see the metal rods in the feet.
Yeah, and so there's some that are a mix, but we also have some that are a little bit mummied and have fabric over the faces. So we pulled out a really nice looking one for you guys.
He's really beautiful. The color is amazing.
Yeah.
And this scanner preserves the color data about the surface.
Yeah, that's one really nice thing about some of these scanners is that a lot of the scanners nowadays are actually like preserving color really well and preserving texture. And so we have it on an HD mode and it's structured light scanning, 3D surface scanning. So we're actually able to pretty accurately pick up the color. We had some trial and error and ended up having to put up black fabric so it wasn't reflecting light in a weird way, but that ended up helping.
And this is useful to researchers. They can go through the digital file and look at the color detail at a really high resolution.
Yeah, and on the platform that we're sharing it on, they can actually measure too. So they can look at the color, they can look at the texture, and they can also measure the specimen if they want to as well.
So this gathers all of that data. It doesn't scan anything inside, below the surface of the scan.
Correct, so that's where you would come in and use CT, but for these specimens, because they have the metal rods inside, and because of the size as well, we're not really able to CT scan these.
There's so much personality in his face.
I know. This one I really love.
Really?
We have a sign on our table that says no gloves, no boop, because it's really hard not to boop their little noses sometimes. But
There's no booping. I love that. Do you ever give names to these guys, or do they have names attached to them at all?
we have some of our 3D prints.
Oh!
And
these are from this.
Yeah, so we have been trialing 3D printing some of our specimens. And so this one's Gus. And this one's Danny.
Amazing. Oh, and the detail is incredible.
Yeah, and so you can see it is actually able to pick up the texture as well.
Right.
And so then our education center can use this. Our docents can use this on the main floor. Mm-hmm. So people can kind of get an idea even at a small scale.
Is this ever useful to researchers to be able to 3D print to these scans as well?
Yeah, actually, we've also 3D printed some skulls too.
Oh.
And so these skulls go hand in hand with the tortoises. So these are actually the real size of the tortoise. And then this is the size of the skull for that tortoise.
Oh, amazing. That's really cool.
And it's a great way too because, you know, people from the public can't actually touch the specimens because they may be prepared in a certain way or covered in arsenic. But with these, with the 3D scans, they can actually like physically touch them and see and get an idea.
That's really thrilling. Walk us through the scan.
Yeah, sure.
This is so cool.
Yeah, so typically I would start with it on its upper side and get the majority of it. But we'll start it on its back side.
Okay.
And this allows me to track where it is.
Oh, like to make sure you've covered everything.
Right. And so then I just start the scan out. And then I start it. And then it allows me to kind of track where I'm going.
Wow.
And it's just collecting hundreds of images with the texture, the color.
After you've spent a whole day doing this, do you end up feeling a little like you're the Terminator, like you're scanning everything, running it through your database?
Not necessarily, but I have a lot of fun doing it because it actually really is cool to see it on a screen, a computer screen.
Yeah.
And so it's fun to see it, you know, in computer programs now you can add a human to it, so you can kind of see
Its scale.
a scale for it as well. And so now with technology, it's really fun to actually see what it might look like if you were to see them in the wild for people.
I love the fact that just by doing this, you make this accessible to anybody with a computer
Right.
anywhere in the world.
Yeah, and so there's two platforms that we're uploading on. One is more research-based, and that's from Morphosource. And then the other is called Sketchfab, and that's a platform where we're really trying to allow education and public to view these scans, and they can use it in their classrooms, or if they have, you know, after-school programs that they're using, they can access these scans and do whatever they would like.
Do you guys build, you must have some curriculums that you build on some of these scans for educators?
Yeah, so part of the grant that we're on is going to be creating a module once we have these scans. And so people will be able to use these in their classrooms.
I mean, looking at the color on the shell, on this carapace, I see all these layers. Is this a way, it just occurred to me, can you tell how old the turtle is by counting the rings?
The data that we have from these specimens are georeferenced, but some of the information we have, just because a lot of these tortoises are from the 1905 and 1906 expedition, some of the information isn't as detailed as what we might collect in today's field work.
Sure. How long does the full scan take for an object this big when you're doing it for real?
For a full scan, no matter what size, it takes me about 10 minutes. Honestly, the longest part is maneuvering the tortoise because we have to be so careful with it. And so then I can show you. You can see that it would pull it up in color. And then you can actually check. So like if there are a lot of areas that are pulling up in red, then that would tell me, oh, I need to actually go back and try to fill in those areas so that when I'm actually aligning the scan together, I'm not having gaps in those sections.
That's really neat.
But then you go in, you delete the inflatable, and then you would merge the different scans together.
Oh, that is so freaking cool.
But you can tell it picks up the texture pretty well, too.
Yeah. Yeah, there are ways you can get closer on this digital scan than you can almost get with your eye. Kate, that is really thrilling. Thank you so much for walking me through that.
Adam.
Tell me what we're about to look at here.
Are you ready?
I am so ready.
So, okay, this is our show-off tray. I have to say, it's a little fancy, but this is a male and a female of each of the species of finches from the Galapagos. So most of these specimens were collected in 1905 and 1906 by Rollo Beck and his team that went down to the Galapagos. And they collected all of the birds that we have, most of the birds that we have from the Galapagos. But these are really helpful in studying natural selection. Right, so these are relatives. They had one common ancestor. but they've evolved to live in different places on the Galapagos.
And these are from 2 different locations.
2 different locations, different feeding strategies, different body size to live in different places. So great examples of natural selection. And the Galapagos is a great place to study natural selection. So there's one of each.
I have a question. When Darwin, in 1905, when you guys were collecting these specimens, was it understood just on what kind of timeline that differentiation had happened on yet?
Whether the evolution, well, Darwin's theory of natural selection had come out prior to 1905. So did Rollo Beck know? I don't know. He was really a collector. He collected, I think, over 25,000 birds in his lifetime. It was a whole different collecting mindset back then. Like, his job was to get hired by museums and travel around the world and collect specimens.
So he was just
gathering. Did he know Darwin's theory of natural selection? I don't know, maybe.
Yeah. But these guys were on a boat heading back to San Francisco when the collection here disappeared.
That's right.
And so for a while, the whole collection was just on its way to San Francisco.
That's right. So in 1906, when the fire happened after the earthquake in San Francisco, everything in the Academy's collection was destroyed, including our journals. So we don't even know what we had. The catalogs were all burned too. So the 8 men in the Galapagos heard about the fire, and they knew they were the new museum. So they brought back over 78,000 specimens, about 6,000 birds.
Yeah.
Lots of plants, lots of insects, and of course the herpetology specimens as well.
I'm looking at the beautiful handwriting on each of these tags. So would this have happened in the field or once it got back here?
No, they wrote that in the field.
Okay.
So that was Geospiza magnorostris from Abingdon in the Galapagos, September 22nd, 1906. Wow. That's the female. And then you can see who the collector was.
Oh.
And there's R.H. Beck, that's Rollo Beck. He was the leader of the team, and he was also a big supporter of the Academy for many years. He lived in Pacifica and would go out in his little dinghy and collect seabirds off of California.
Really?
And he actually held the record for the fastest bird skinning, right? Because they would collect the birds and then they'd have to prepare them in the field.
That's my other question. Yeah.
So bird skinning, for these skins, you make an incision down the breast and you turn it inside out like you're taking a sweatshirt off over your head. And then we take most of the insides. The skull is still there, the wing bones are still there, and the leg bones are still there. But all the soft parts come out, so it doesn't rot.
Right.
I'm wearing gloves because they used arsenic. So they had arsenic soap that they rubbed on them. And then they just stuffed them full of cotton, so you can see they have little cotton eyes. Wow. And then let them dry. And we still do this today, minus the arsenic, of course.
Right.
And
the arsenic was a useful preservative back then?
Yep.
Okay.
And it does work to keep pests away. So they've survived from 1906, 1905, 1906 to now. So over 100 and almost 120 years. So.
And you guys are you are digitizing these as well.
We are. So some of these birds are actually now surface scanned. So different from the CT scan work that you saw with the Galapagos finches and the tortoises and the iguanas in alcohol.
Yeah.
Because these are empty inside, there's no point doing a CT scan.
So there's not even any real skeletal structure?
Well, there's just part of the skull, the wing, and the legs.
Got it.
Well, you can see the legs. They're all right there. So they're really more useful for surface scanning, so an exterior scan that documents the measurements. So researchers who can't get to this collection, it's hard to loan out these specimens. The Galapagos birds don't go on loan like the rest of our collection, so researchers have to come here to study them. But if you can't get to the collection, you can look at these surface scans and you can get measurements of the bills. You can get measurements of the tarsus, which is a leg measurement that helps scientists. They measure this bone right here, toe measurements. So you can still get the measurements from the surface scans. And it just makes the Galapagos skin specimen more accessible to more people worldwide in those scans.
the beaks are a very obvious differentiation, but I'm curious if there's also a differentiation in feather types. Is that also something that you guys chronicle or document?
That's not captured as well on the surface scanning.
Sure.
The technique that we used. Researchers have come and sampled feathers, and this is a whole different area of research, and you wouldn't be able to get it from the digital world, but people have come to study mercury on feathers. You can look at stable isotopes in feathers, and it tells you where an animal was while it was growing that feather.
Fascinating.
So you can look at the chemical analysis of the feathers. Researchers have also come into our collection to look at color in feathers. There was a researcher at Stanford who used the slack he put feathers at the end of the slack, the Stanford Linear Accelerator, and then shot something at them, laser rays or something, to look at refraction off of bird feathers like that. So things that researchers can do with these specimens, back in 1905 and 1906, they had no idea what would happen 100 years later. Like, we didn't know about DNA until 1953. They were just collecting these birds, preparing them, and building the Academy's collection. That was their main goal. They were probably eating the insides of the birds. I mean, they had to eat something, right? If you caught it, you might as well be sustainable. I don't know that for sure. I just expect that they did. If I was in the field, that's what I would have done back then.
When you're digitizing these, are you also digitizing the labels? Do those travel with the scanners?
The labels go mostly with, not so much with the surface scans. They kind of stick out on there, but they're not formally labeled.
I look at those and it makes me feel like there's a person there. Like
Rollo Beck is sitting there in his little tent, writing the label on the tag.
That's amazing.
And then the new catalog number is from here.
Okay.
It gets the catalog number here. So these are the first thousands in our collection, right? Because we had to start at zero again.
Right. Oh, of course it is.
Yeah. So this is the show-off tray. Let me show you what a regular tray looks like.
Oh my God.
Right, lots of individuals of different species from different locations. And it's really important to have that kind of diversity so you can really see the differences in individuals and in species. Like, if we took 2 kids out of a 3rd grade class in San Francisco and said everybody looked like those 2, we'd be wrong. So that's why we need larger sample sizes in science.
And you also do surface scans of these too, so researchers can understand that.
Right. So we surface scanned several of each species of birds that came from the Galapagos.
Okay.
So I think we did over 400 surface scans of the birds in this collection. And that's just the tip of the iceberg of what we have. So we have 60% of the cataloged birds in museums that we can search the catalog. There might be some out there that are not in an electronic catalog that's searchable that we don't know about. But of the catalogs you can search.
You have 60% of the specimens collected from the Galapagos. That's incredible.
So a researcher could come here and have a plethora of individuals to study. Peter and Rosemary Grant, David Lack, all the big people who studied evolution came and used this collection and the other collections as well in the Netherlands and in the British Museum too.
Now, when the expedition was in the Galapagos in 1905, are they setting up traps or nets to just willy-nilly collect birds?
Nets we use nowadays. Back then, they used shotguns, and they were really good with shotguns.
Really?
Yeah, so some of the CT scans sometimes will have shotgun pellets, because for birds, you have something called dust shot, which is really like sand almost. It's very small, so it doesn't damage the birds. So it was a whole different world back then. We don't do that kind of collecting unless we have a specific project in mind nowadays. Nowadays, we add to the collection through salvaging, birds that hit buildings, birds that are found dead on beaches, birds that die in a rehab center or found on roads. But back then, they didn't have binoculars. The only way to really study birds in the early 1900s was to hold them in your hands.
Catch it, kill it, study it.
Yep.
People come to museums, we all come to museums as school children, and we think of them as chronicles of what is. But what I love is what a living, breathing research facility this is,
Right.
and accommodating the fact that all of your ideas about how things are might change also in the future, as it has
Exactly.
changed from 120 years ago. to now.
Exactly. This is a record of what life was like in the Galapagos in 1905 and 1906 for these species. You can look at the nests that we have from back then, also from the 05 and 06 expedition. I can show you some of those.
Yeah,
And you can see the plants that were used back then.
of course.
Yeah.
So you get so much environmental data from these as well about the local conditions.
Yep. And with the stable isotopes and what you can find inside the species that we never knew, we could find in 100 years, there'll be something else that our ancestors are studying, and they'll be grateful that we kept everything here. So a lot of responsibility on us collection managers and the museum as a whole to make sure that this material is here in the future for people to study, for whatever they can dream of, whatever questions come to mind.
I'm just so, these labels are so thrilling, and I'm picturing boxes of tens of thousands of them leaving here in 1904, going to the Galapagos. There's something so, is there a drawer here that like thrills you every time you open it?
Oh, I mean, the Galapagos
Yeah,
finches
it's just
are really
everything,
fun. Everything, like when we recently had to do like your favorite specimen.
Yeah.
And I can't come up with
very
any,
different.
it's so hard. We have 100,000 birds, 35,000 mammals, and 11,000 eggs and nests. Like, how can you pick a favorite out of all those? But it just inspires me every time I share the collection with people, whether it's a tour of college students coming through, or donors, or people like you who just want to are interested in the collection. Every time I share the collection with people, that's really what thrills me. Like, I take care of this unique library of dead things, and it makes me proud.
It is really thrilling. You said there are some nests?
Yeah, let's see if we can find the nests.
Look at nests.
So we have bird skins up until this point in the collection. And then we have bird skeletons. And then the egg and nest collection starts. So we will have to roll this compactor.
I need this kind of thing in my life someday. I love this so
The
much.
only problem is if we have like 2 researchers visiting, you have to plan it so that they're not in the same row.
Yeah, like a dance.
Yes. Moving in was really fun. So these are some of our Galapagos nests. They're amazing. They're absolutely amazing. So here are some of the birds that we saw upstairs or over there.
Oh my goodness.
So there are those labels again,
Oh, look at that.
only now they're on. And then you can see the native cotton. And what's interesting about Galapagos finches is they build a little roof on their nests. Not many birds.
Oh, so this is upright.
Yeah, so
Okay.
it sits upright in the tree like this.
They like a little protection from the rain.
Well, and the sun, the equatorial sun, right?
Oh, of course.
Makes total sense. Yeah. So I don't know how they got these all back without.
Isn't that that's
I know.
astounding when you think about temperature control, and did
I
they
know.
go around the horn? Yeah, of course they came
I
around the horn.
assume,
The
yeah.
Panama Canal wasn't there.
Yeah.
Crazy.
There's another one of the geospiza nests with the cotton balls in it. So that's pretty substantial.
It really is. They're in insanely great shape. It
So-
would seem.
Yeah, yes, for 100 and plus years. We had a researcher come not for the Galapagos, but for a local China camp restoration project where they're looking to restore the environment for species. And they took samples of nest material and then did genetics, they didn't find the endangered extinct rows in our collection, but they did in a nest from Berkeley. So they came here and sampled the little sticks from the nest to see what was being used.
That's amazing.
That's no longer there anymore.
Yeah.
So they might bring back those extinct plant species too, in addition to helping the bird species that are endangered. And then here's some eggs.
tiny
Yeah.
eggs.
This is from Chatham Island in 1906. And it was field number 607. And then they used to, this was Hunter, he was one of the ornithologists on the trip.
So
Built of lichen and grass, a few small twigs about 10 feet from ground, lined with something
lichen.
and cotton balls. And those cotton balls are not the cotton balls that we know.
Yeah,
it's
yeah.
the native cotton in Hawaii. I mean, in Galapagos.
To gather up 70 some odd thousand specimens, how long was the expedition in the Galapagos?
I don't know the exact dates because I know they did some collecting on the way down and then they were there and then they did a little bit on the way back, although they came back much more quickly than they probably would have because they knew that the museum had burned and they had to ensure that everything came back safely. And it was really dangerous to go into port in your wooden boats at that time because bad things could happen in port. So
and they were
came,
protecting now and a completely irreplaceable resource.
A whole new museum, basically. And then the Academy schooner was in the bay for, I think, 2 years as that was the Academy.
That was the actual museum.
Yeah,
oh
and it was called the Academy. The schooner was called the Academy. And now there's Academy Bay in the Galapagos too.
Yeah.
So,
this is a movie. I totally want to watch this series about this journey. It sounds incredible.
Rollo Beck and his other 7 sailor scientists. Like one of the things that inspires me too about this collection is, they were 8 men in the Galapagos and a lot of women use this collection as scientists now that I'm sure that they couldn't even
never
fathom
occurred to
that
them.
either. Like 100 years from now, a female scientist is going to look at the lesions in the feet of the birds that we're skinning.
Yeah.
No. To study the virus, not to study the bird.
Right, right.
Never would have occurred to them. So
About how much the collection could teach future generations.
exactly.
That's really lovely.
They probably had some, their goal was to collect and to build collections. So they had an understanding of collections, but certainly not in the same way we do now with technology as it's advanced over the years.
Well, thank you, Mo. That was
Absolutely.
really, really thrilling.